that recording. So that way this is up here as well. So I want you guys to work with me on this in creating these graphs and talking about these two different types of cells so that we're kind of reviewing it together. I mean, we're not going to bunch it out to like think, pair, share exercises or anything like that right now, but we could do that in the future if you guys wanted to have some time to discuss these things amongst yourself when we get together. We could add that in sometime coming up too. But what I'm going to start with is we're going to be looking at and comparing the two different types of cardiac muscle cells. So I already mentioned them. What are the two different types of cardiac muscle cells? So we have one that starts with an A. So it's called autorhythmic, right? So they're creating this beat. They are the bosses. They're the conductors, right? So we have autorhythmic cells. And then the other ones are doing the actual work. They're doing the actual job. So those ones are going to be, instead, contractile cells. Yeah. Okay, so that's the first thing to lay out, is we need to know that there are two different types of cardiac muscle cells. We didn't know that before. Now we do. So with the contractile and autorhythmic cells, one of them is all over the place. We have a lot of them. One of them we have very few of. Which are the ones that we have a lot of comparatively? So contractile cells, right? So we have, that's like 99% or more of all of our different cardiac muscle cells are going to be contractile cells, whereas autorhythmic cells are going to be about 1% instead, okay? In terms of where we can find them, the autorhythmic cells, that there are not very many of, they will only be found as part of the conducting or the conductive system of the heart. So they are going to be found in areas like the SA node and the AV node and the bundle of His or the bundle branches and the Purkinje fibers and stuff like that. There's that one figure where it actually shows you these kind of yellow lines going through the walls of the heart that kind of look like nerves. Not nerves, but autorhythmic cells. So any of those places where we're seeing the part of the conductive system of the heart, that's going to be made up of our autorhythmic cells. Yeah.
Are they linked together then, or are they individually on the cap?
They are linked together for sure. So they will have like intercalated discs and stuff like that connecting them. Though their morphology, their anatomy is a little bit different than the contractile cells because they have a different, job. So in a way, they can be a little bit longer because, like a neuron, right, it has to go longer distances, have more connections, and things like that. The funny thing about that, though, is that a number of years ago, I was thinking the same thing. And again, it's not going to be on a lecture exam or whatever. You're not going to have to physically identify. We don't have two different cardiac muscle slides, right? We just have one. And most of what you see in that cardiac muscle slide, which half of you have seen already and half of you will see when lab comes up, is we're looking at those contractile cells, right? With the stripes or striations that show the overlapping actin and myosin, just like our skeletal muscle cells. So textbooks and stuff like that, for whatever reason, they don't show you what the autorhythmic cells look like. You're like, well, we're talking about it, and we see they're located in these different places, and they have different jobs. How come we can't see them? And I've looked around, and I've asked around and stuff like that, and I haven't found anybody who says, yeah, here's some good photos of autorhythmic cells. But, again, if we think about function and form, we would think that since these are acting a bit like neurons, where they have to have a bunch of connections to communicate, they have to go kind of longer distances to be able to extend to different areas, they probably have more of a morphology tending more towards neurons compared to our striated contractile cells. But they don't show us that.
So you said that they kind of go on their own, but what stimulates them to just go on their own? Like the neurons obviously are stimulated by the brain.
Mm-hmm.
So what stimulates the heart to just keep going and the autorhythmic cells to keep going?
Yeah. So two aspects about that. So with the autorhythmic cells generating their own rhythm, they'll generate their own basic rhythm as well. Now that basic rhythm, we're going to come into when we talk about these graphs as to why, and here I'm just doing our axes like usual, as to why they will kind of do this, again, send a signal and then go back down, and send a signal and go back down like that. It has to do with, if you remember when we're talking about any other types of cells, when we're talking about cell membranes, we have different types of channels. We have leak channels, and we have gated channels and stuff like that. So leak channels are continuously, like a small hole in your bike tire, going to be letting stuff out from the inside to the outside, or in other cases from the outside to the inside, right? So letting things cross the membrane. And gated channels are going to require a signal, an electrical signal, a chemical signal, or something like that to tell them, oh, it's time to open, oh, it's time to close instead. So we're going to find that things like leak channels in our autorhythmic cells will actually be continuously kind of depolarizing and getting us up to threshold where then our voltage-gated channels are going to respond and open up. So because we have that slow leak like that, it's continuously getting us to generating another action potential again and again. So rather than sitting at resting potential, and that's one of the things we're going to compare, we don't see a flat line. We don't see a resting potential where, okay, we're maintaining this balance like this until something happens, until we get stimulated. They're constantly changing instead. Yeah. Okay, so autorhythmic cells, we'll start with that then. With autorhythmic cells, as we mentioned, we don't see any area where our membrane potential is flat. So we don't really see a resting potential like we did with skeletal muscle cells where, ooh, it's relaxing, it's resting, it's not doing anything, it's just a flat line in terms of membrane potential. So what we see is that it's constantly changing. So there is a period of time where it's a slow depolarization like that, okay? So these are negative voltages, like about negative 60 millivolts or so. Remember, this is time on the x-axis here. And so there's a period of time where it's doing a slow depolarization. So that is where it's becoming, maybe 0 millivolts like that, it's becoming less and less negative. That slow depolarization is due to those sodium channels, right? So sodium is an ion that is always found in higher concentrations. We don't want to say always, mostly found in higher concentrations outside the cell. So if you have a channel that's open, sodium will flow from where it's high outside the cell to inside the cell. So when the sodium goes into the cell, it's going to make it less and less negative because sodium is a positively charged ion, right? It's a cation. So this part, this slow depolarization, is due to sodium leaking into the cell, making it less and less negative. Now, at some point, there's going to be a threshold here. And when that threshold is reached, then we're going to have a fast depolarization. So we see that curve get much steeper. And that's going to be due to voltage-gated calcium channels. So these calcium channels are gated. They're waiting for a particular voltage of the membrane potential. And when it reaches that, now all of these calcium channels open up. So now positively charged calcium, because we have a bunch outside the cell, is going to flood into the cell, and that's going to make it depolarized even more and faster. Because not only do we have all of these channels opening up at the same time, but calcium has a +2 charge. So for every one calcium ion that goes in, we have 2 plus charges that are going in. So that's depolarizing it even faster. Okay? So when we get up to about 0 millivolts or so, we're going to see a change. We're going to see it start coming down again instead. And so that, remember, all this is depolarization, this is repolarization. So how do we repolarize it? We repolarize it, and again, these things have been going in and in. Now we have potassium ions that are going to leak out of the cell because potassium ions are maintained at higher concentrations inside the cell. So if we open up these voltage-gated channels, and we do so at about 0 millivolts, when we open up those channels, those positively charged potassium ions that are in high concentration inside the cell are going to diffuse out. And as we lose positively charged ions, that makes the inside of the cell more and more negative, right? So like in terms of your own mood, if you're losing positive things out of your life, oh, I got a flat tire today, and I got bills due today, and they were out of my favorite drink at Starbucks, that's all, you're losing all these positive things, so your mood becomes more and more negative, right? That's what's going on in the cell. We're losing these positively charged potassium ions, making it more and more negative again, okay? Then that comes down to here, and now as those potassium channels are going to close, now that leaking sodium is going to start taking us to threshold again. And now it's going to do that again. Get up to certain thresholds, we have calcium channels opening, and then potassium channels opening, and now we start going again. Okay? So we're going to see this continuous pattern of depolarization, repolarization, and now we just start depolarizing right afterwards again. Okay? No flatlining there in these autorhythmic cells. So we see slow depolarization due to sodium, faster depolarization due to calcium, and then repolarization due to potassium. Sodium, then calcium, then potassium. Okay? And this is our autorhythmic cells that are found in places like the SA node, the AV node, stuff like that. And they are creating this tempo, this heart rate. Okay? They're conducting the contractile cells, telling them when they're going to contract and things like that. Okay? So this is like a drum beat. We're generating the rhythm. Everybody else is going to be following that rhythm. Questions on that? Okay. Now we move over to contractile cells. So contractile cells, these are going to be found in the middle layer of the heart, where all the heart muscle is, right? So it's going to be found in the myocardium, all over the place, lining the walls of the atria, lining the walls of the ventricles. That's where we're going to find contractile cells. They're going to be squeezing those chambers and ejecting blood as necessary, and then relaxing and filling again. So these ones will, in fact, have a resting potential. They need to relax to allow those chambers to fill again before they contract again. So we have a prolonged relaxation or resting period that we see here. And again, time here, and millivolts over here. And then what happens is they're going to get their signal. They're going to be told by our autorhythmic cells to contract. So what's going to happen is we're going to have a bunch of sodium channels. That's nice. Sodium channels that are going to open up. And they are going to let a bunch of sodium go in, flood into the cell. So we have a very, very steep depolarization period in our contractile cells. So when the contractile cells finish depolarizing, what we're going to find is they're not going to come straight back down. They have this flattened period like this before they start coming back down. This is called the plateau. And the plateau, ooh, I reversed those letters. It's an AU like that. The plateau is due to calcium channels, keeping it in this depolarized state. So once we get up here, our sodium channels are going to start closing, but now we have voltage-gated calcium channels that are opening, letting this positively charged calcium go into the cell as well, keeping it in this depolarized state. Okay? Now once these calcium channels close, now potassium channels are going to open. We know potassium leaks out of the cell because it's higher concentration on the inside. That's going to repolarize it. And then we go back to our resting membrane potential again. So we see the same ions involved. First sodium, then calcium, then potassium. But in this case, sodium does our depolarization. Calcium maintains our depolarization in a plateau. And this lasts for like a quarter of a second or so. Depends on the textbook. They'll say 250 or 300 milliseconds or something like that. So a quarter to a third of a second, which is a long time. It will maintain it in this depolarized state. So these muscle cells will contract for a long period of time, and then they'll repolarize and get set to go again. Okay? So same ions in the same order, but just kind of prolonged as well. Okay? And we do have a resting potential there. We don't really have a resting potential here. Questions?
So does that mean that the sodium channels are not leaky in the contractile?
Right, yeah. So these ones are actually the gated ones that are opening up very, very quickly when these cells are getting stimulated by our autorhythmic cells. And so that's why we have such a steep curve here, is because these are going to be our gated ones that are opening up very quickly. And they will, like we said, the gates will close up here at this membrane potential, but now we have calcium channels that are opening up, and they'll keep it in that depolarized state. So, and then when we get to here, we're also going to have potassium channels opening up. So this is kind of our transition where we have, oh, there's some of these positively charged calcium coming in. Now we're starting to offset with some positively charged potassium leaking out. So now we started to see a little bit of that drop. We'll see calcium channels are going to start closing. That's when we see more of a precipitous drop, more of a steep drop. Calcium channels are closed. Now it's just all this potassium leaking out instead. Okay? So we have a prolonged action potential in our contractile cells. That's going to give us a long squeeze, like we said, like squeezing the sponge for a long period of time to allow that fluid to get out. And then we have a long, what's called refractory period after this. So there's a refractory period when our cardiac muscle contractile cells cannot respond, will not respond. Even if you hit them with more stimulation from our conductive cells, they're not going to be able to respond. And that's because we need them to contract for a long period of time to squeeze that fluid out, and then they can relax. If we contract and now we just relax a little bit, that's preload. We only preloaded it a little bit. That means next time you squeeze it, you're not going to get a lot of blood out. So it's really important that we have a long squeeze and a long relaxation and filling period, so that next time we squeeze, we have a lot of blood that's going to be able to go out. Okay? Yeah.
So the action potential is where it's ejecting the blood. It's contracting to eject the blood, and then the refractory is when it's filling.
So that's a good point as well. So what we're looking at here is really electrical activity. We're looking at changes in membrane potential by the movement of these charged particles across the ion of an individual cell. In terms of the mechanical response, like, oh, well, when is this cell, if we're talking about a contractile cell, when is it actually going to contract and things like that? It happens, almost instantaneously after that. And so we're going to get our contraction occurring right after this action potential initiates, and then our refractory, where it's going to go into a relaxation phase, is going to be kind of shifted. It's going to be like here instead. So much like when we're doing the EKG, when we see that, we have our P wave, which is where the atrial walls are getting their signal, and it's right after that P wave where they're going to contract in response to that after the atria depolarize. So the mechanical response, the contraction, occurs right after the electrical stimulation, right? So even if you had like a muscle stim unit or something like that, if you're doing physical therapy and they put muscle stim on you, when that unit sends a signal, the signal goes at this time, and there's a fraction of a second, the time later when the signal arrives and the muscle actually responds, right? Even like your brain, right? When you're talking about picking up this pen or something like that, it seems instantaneous, but you had to generate the thought first, the neural pathway, right? The motor neuron had to send the signal, and then the muscles responded. So there is a kind of a delay, and the same thing here. So the actual response, the mechanical contraction, is going to be just kind of shifted just right after we see this action potential occurring. Okay? Okay. Are there other questions about heart stuff? The cardio part of cardiovascular. So with these, comparing the membrane potentials and the action potentials of these two types of cells, you want to practice it, right? So create a list, like what are the two cells? Where are they found? How many do we have on a relative frequency? What are the ions involved? What's the ion involved in depolarization and repolarization and stuff like that? How long is the action potential? Not in terms of actual numbers, but right? The action potential in contractile cells is much longer. This is still on the order of like 5 to 10 milliseconds. And this is on the order of, like you said, like 250 to 300 milliseconds. Where can you find them? In the myocardium versus in the SA node in the conducting system of the heart. So whether you draw them out like this or whether you just kind of describe what's going on in each, doesn't matter, but you want to practice with it. So you'd be able to do your own compare and contrast if you have questions on it about the exam. Yeah?
I have a question on the heart objectives, I think it's number 13, says explain the function of the fibrous connective tissue septum, which separates the atrial and ventricular myocardium.
That's a good question. So that one, let me just briefly open up that heart PowerPoint. Because when we were going through the anatomy, we kind of jumped a little bit through the anatomy because we said that we were covering it in lab, which we did. But this shot that we see right here shows us a transverse section through the heart, and it shows us all 4 valves. Remember, we have atrioventricular valves with the chordae tendineae, and we have semilunar valves. So on a particular section, if you cut it through, you can actually see all 4 of them in the same plane. Well, all of those valves are embedded within what's called the fibrous skeleton. So it's like almost like a dense regular connective tissue, like a tendon, and it serves as kind of an anchoring point for all of those different valves. So we said that those valves are kind of like doors, and in order for a door to work, it has to have a very stable door jamb with the hinges on it, and that stabilizes the door. So you can always find the door moving within the door jamb like that. So the fibrous skeleton of the heart is a bunch of connective tissue that runs right to the middle of it. It anchors all of the valves, and a lot of our cardiac muscle fibers anchor there as well. Because rather than skeletal muscle where it's like, oh, it attaches this bone to that bone, and when we pull on it, those bones move relative to one another. We have these bundles of cardiac muscle that will anchor on this connective tissue instead. So it serves as an anchoring point in here, in between here, for the valves. It also serves as an anchoring point for the muscle tissue. But the other key thing about it is that when we're looking at this conducting system of the heart, we said that the atria, it's very important that the atria get the signal first and then the ventricles, right? Because the activity of the heart is not all squeezing everything at the same time. It's atria, ventricles, atria, ventricles, atria, ventricles, right? So we want the atria to get the signal first and then the ventricles afterwards, not at the same time. And so when that signal is going through our autorhythmic cells, starting at the SA node and spreading to the atria and then the AV node and then spreading down the bundle to get to the ventricles, it has to go through that connective tissue there. And when it goes through that connective tissue is not very conductive, and so it actually slows it down. And so the signal gets slowed down between the atria and the ventricles so that the atria can get that signal first and start to respond before the ventricles get their signal. It gives us what's called an AV nodal delay, so that we make sure the ventricles gets the signal afterwards, and we're not locking the whole thing up. Okay? A bit, yeah. Okay? So we already knew that it was important for the atria to get the signal first and then the ventricles so that we can work back and forth. That's part of how that signal is delayed that way, so it doesn't just zap the whole heart at the same time, right? Atria then spreads down to the ventricles. Okay. All right, so we'll keep thinking on heart, and if we have more heart questions, we can jump into those at the beginning of next time as well. But what I'm going to do now is introduce us to our blood vessels, and that way next time as well, we can go over those two if we need to, and keep moving forward. So the vascular component of cardiovascular system is what we're going to be looking at today, and like usual, the way we have this organized is we're going to be looking at big picture, then we're going to kind of get down smaller, and then we'll kind of zoom out again. And then the second half of this lecture talks a bit about what's called hemodynamics. So it ties together, okay, the heart is the pump that moves the blood. We get that. We talked about how it does that in sequence and stuff like that. The blood vessels are going to be the pipes that conduct the blood around. But we have to understand about blood pressure and blood flow and stuff like that, which is critical to our future patients. So that's kind of the second half of the lecture, is talking about hemodynamics and blood pressure and blood flow and how it's generated and things like that. Because it's a big loop, right? This is a closed cardiovascular system. It begins and ends at the heart, and then we have these different blood vessels that are, like I said, the pipes that are routing this blood around. And when we talk about changes in blood pressure and blood flow, it can certainly be due to changes in the heart, right? Because we said that cardiac output is affected by heart rate, how fast it's going, as well as stroke volume, right? How much you're getting out of individual squeeze. So changes in the heart activity can affect blood pressure and blood flow, but also anything that happens out in the blood vessels can help that as well. So if a blood vessel changes diameter, if it constricts or dilates, that also is going to affect blood pressure and blood flow, right? So just like if you're talking about your house and you're thinking about the pipes that you have there, if you have a pump in some way, right, generally our water pressure is coming from the street and there's high pressure that's delivered by the town or whatever, you don't have a pump. But if you lived out on your own, you might actually have a well that has a pump. If the pump goes out, no more water is getting delivered, right? That's going to affect the delivery at the spigot, right? So certainly if the pump, anything that happens with the pump will affect fluid flow. But also if something happens with the pipe, if you have a pipe break, if you have a pipe get clogged because your kid flushed a toy down the toilet or something like that, that also is going to affect water flow, right? Fluid flow, because it's a problem with the pipe now. So all of these things are connected in the body as well. So if there's a problem and you have a patient that comes in and they've got problems with blood pressure or fluid flow or fluid is getting stuck in a limb or something like that, you have to start looking. Is there a problem with the pump? Is there a problem with the pipe? Is there a problem with the fluid as well, right? So same thing, if your house, if the well is going dry, or if something got into the well and now it's getting contaminated with something, that also can affect the system. So there can be problems with the blood as well. If you're dehydrated, if you're overhydrated, if you have anemia or something like that, that also can affect fluid volume and fluid pressure and stuff. So it all kind of ties together. Okay, so when we're talking about pipes though, we have our different vessels. Now, the three major categories of blood vessels are going to be arteries and veins and capillaries. But there are going to be kind of subcategories as well. Remember how when we were talking about blood cells, we said, oh, basic types, red blood cells, white blood cells, platelets. But then as we got into it, we're like, wait, white blood cells, it's 5 different types of white blood cells, and we need to know structure and function for each of those, right? That's another exam objective question. You need to know it for each of those 5 different white blood cells, as well as what platelets do, as well as what red blood cells do. So that's 7 types of cells. So here, 3 basic types of blood vessels, but we'll have specialized subtypes as well that we'll be coming up to. And like with the blood cells, we need to know, okay, why do we have all these different types? What's different about them in terms of their structure and their function? If we have different types, there's got to be something different about them. So already we've got arteries and veins. What's the difference between an artery and a vein in terms of their function? Arteries are always taking blood away from the heart. Veins are always taking blood back to the heart, right? Always. So we're going to see differences in terms of their structure based on that, right? High pressure blood coming from the heart going into these arteries, as opposed to veins are all the way around the other side of the loop. It's been a long time that blood has been circulating around by the time it gets back to veins. So low pressure blood there. So we'll see some changes there. Capillaries are really important because these are the exchange vessels. So all of our other larger vessels is just about getting it from one place to another, kind of like the canals that we have going through Phoenix or something like that, right? So it's just about routing it to different places around town. But at some point, we want to be able to exchange with tissues. So in the blood, we know that in the plasma and in those cells, we've got a lot of critical things. We've got oxygen, we've got nutrients, we've got water, we've got ions. All these things we need to give to the tissues to keep them alive. Not only do we need to get it into their neighborhood, like, hey, great, I reached the bottom of the foot, but I need to get it out of the blood vessel and to the cells that are in that neighborhood, right? So it's one thing if the Amazon Prime truck comes into your neighborhood, you're like, oh, it's coming, I'm tracking it. And if they just zoom through without delivering it, then you didn't get your package, right? So same thing in terms of cells. We've got to have these capillaries where things can actually be delivered from the blood into the tissue. And so capillaries are small, super small, very thin blood vessels where exchange occurs. Things go out from the blood and even from the tissues back into the blood itself. Okay. So the capillaries are going to be our exchange vessels. They're going to be the most numerous. They're going to be the thinnest ones as well. So this diagram that we see here shows us a system, a circuit of blood vessels. This is showing us a bit of our systemic circuit. So we see our arteries in red and our veins in blue. There's our heart. So we see coming out like this, we're going to have our major arteries. Now notice there are elastic arteries and muscular arteries. Oh, so right off the bat, we've got two different types of specialized arteries. So we're going to have to talk about those. After we get through these arteries, we know arteries as vessels, as they go out, when they branch, they're going to get different names and things like that. So we're learning that in lab. As we get from arteries, we get down to smaller and smaller branches and more numerous. These are called arterioles. So arterioles are very, very small arteries. And we'll talk about those coming up and why we have arterioles. Arterioles are really the last vessel before we get to our capillaries. And you can see the capillaries, our exchange vessels, are the most numerous, they're the smallest. And notice the color change there. You know, if you're not colorblind, I'm going to tell you that's like a purple. So we started out with a red, designating that we had a high oxygen content. When we get to the capillaries, now it goes purple. That means the oxygen that we were carrying by those red blood cells is being actually delivered to the tissues. And now we're picking up some of their carbon dioxide, but we're also delivering nutrients and picking up their waste products. We're delivering some water and ions and things like that. So there's lots of exchanges going on there. So that's why we see that color change. Now those capillaries drain into small veins that are called venules. So on this side we have small veins called venules. And then the venules end up draining into larger and fewer veins until finally we dump back into the heart, right? So with these vessels, the largest ones are the ones closest to the heart. So for instance here, we have the largest artery of the body, which is the aorta, right? Here, coming back into the heart, we have the vena cava, right? We have the superior and inferior vena cava. All the blood from the whole body comes back to the heart in those two vessels, right? But there's going to be lots of other smaller vessels as we branch out and get to all these different neighborhoods of the body as well. All right, so we're going to be taking a look at that. We're kind of going to do a tour through, as if we were a red blood cell, through our vascular system and talk about each of the different blood vessels that we will encounter along the way, okay? Before we leave this slide, though, I want you to notice right through the center there is some sort of like weird alien system of parasites or something like that inside of us, right? That's what it looks like. It's some weird green color, and I haven't heard of it before, and it's somehow hooked into our system there. So those are lymphatic vessels. We'll address those a little bit in lab as well, but the lymphatic system is a series of vessels that conduct fluids as well. In that exchange that occurs with the capillaries, when we deliver things out to the tissues, we also get things back. That trade is not exactly equal. So in terms of fluids and things like that, we push fluids out into the tissues, right? If you're dehydrated or something like that, you drink more, it gets absorbed across the intestine, goes into the bloodstream, we deliver to the tissues, they rehydrate. So that delivery of fluid that goes out, we do reclaim some of that as well. Remember, in the plasma, we have 3 plasma proteins, right? So one of those starts with an A, has to do with osmotic balance. What's that one called? Albumin, right? So you have to know fibrinogen, you have to know immunoglobulins, which are going to be our antibodies, and then our other major category is albumin. So albumin creates osmotic pressure that draws or sucks water from the tissues back into the bloodstream. So we're exchanging fluid down to tissues, but there's slightly more fluid that goes into the tissues than comes back. And if we didn't clean that up or grab it in some way, our tissues would start to swell. We'd get edema, right? And the fluid would be left there and it would just swell and swell. So the lymphatic vessels, bless you, pick up that excess fluid and then they draw it back and take it back towards the heart. So when they're picking up that fluid, it's called lymph once it goes into the lymphatic vessels. and it routes through these lymphatic vessels towards the heart. And along the way, you can see how there are lymph nodes. Lymph nodes are basically little filtering stations, because when we pick up that fluid, kind of like your pool does, right, there's a filtration system. It picks up a little bit of fluid, and it routes it through a filter, cleans it, and then it dumps it back into circulation. That's what the lymphatic system is doing. Picks up this excess fluid. Before it dumps it back, it cleans it in these lymph nodes, and then it dumps it back up here at the subclavian veins, and now it becomes plasma again. Okay? So that's what that is. Now, lymphatic system and lymphatic and immune system, because it is protecting us against dirty things that it picks up, pathogens, etc. Okay? Lymphatic and immune system is a whole system of the body. It's one of the 11 systems of the body. It's probably the one that people forget about most or haven't heard of. They're like, yeah, I know skeletal system, and I know digestive system, stuff like that. Lymphatic, they're like, is that a new one they just added? So this is behind the scenes. We're going to learn about that after lecture exam 1. We're going to do lymphatic and immune. But we bring it up here because it works closely with our cardiovascular system. So we're just kind of bringing it to your attention. Okay? Okay. So let's take a look at some of these major, just the 3 major blood vessel types, and compare them in terms of their anatomy and their physiology, their structure and their function. So we already have an idea about function. We said that arteries take the high pressure blood away from the heart. Capillaries are the exchange vessels, and then the veins are going to be taking the blood back to the heart. So between our arteries and our veins, and morning lab people, afternoon lab people, or late lab people are like, man, they always get all these previews into everything. But that's what happens. You get up early, early bird gets the worm. So what we find is that with arteries and veins, they have 3 layers to their wall. Okay? They have 3 layers to their walls, just like the heart does. There's an inner lining, there's a middle muscular layer, and then there's an outer layer. Okay? So both arteries and veins have those 3 layers. And those layers, each one is called a tunic. So you see tunica there as part of its first name. The innermost one is called the intima or the interna. You can use either term. Middle one's tunica media, outer one's tunica externa. So arteries and veins both have that. But because the arteries have this high pressure blood coming from the heart, tidal waves of blood that's being pushed down these arteries with every beat of the ventricles, they have to have a much thicker wall to be able to resist that. Otherwise they'll blow out, right? So just like pipes in your house, if the fluid pressure gets too high, the pipe can burst. So definitely those pipes that are coming from your street going to your house are big, thick-walled pipes because they have to resist that pressure. By the time it gets to the spigot, those are fairly small pipes. Sometimes even like PVC pipes, if you're talking about like, irrigation, it could be like flexible plastic pipes because it's low pressure, right? But these arteries have to have a very thick wall because of that high pressure. So even though they have the same 3 layers, the wall itself is much thicker compared to the same 3 layers if we were looking at it in a vein. Okay? Most notably, that middle layer, which is the tunica media where we have smooth muscle, is much thicker in an artery than a vein. So not only to resist that pressure, but also to constrict and dilate to help route that blood to different places where it needs to go. We also notice that the intima, the internal layer in an artery, is very wavy, like you see here, and it's much smoother in a vein. In the artery, we have a lot of elastic fibers that allow it to stretch and then snap back when those tidal waves of blood are coming down each time the ventricles contract, whereas the veins don't need that. There's no more pulsing blood in the vein. So they still have a tunica media, but it's not as thick. It's not as strong as what we see in the arteries. Okay, so that's some comparisons of the artery and vein. Notice that the lumen, the open area, right, this is the lumen, in a vein tends to be much bigger as well compared to the lumen in an artery. Because again, we have that really thick wall that's taking up a lot of that space as well. And in the veins, with such low pressure, we're just kind of eking our way back to the heart. We're just doing whatever we can to escort that blood and get it back to the heart. Because it's been traveling so far and so long, it's tired. It's been dragging along the walls of the blood vessels for a whole minute, which doesn't seem like a long period of time, but it's gone through this whole loop So by the time it gets back there, it's just barely making it back. And so if we open up these veins as much as we can, that makes it easier for it to get through, okay? So oftentimes we see that open lumen, and notice this, valves. So just like in the heart, we have one-way valves so that as it's eking its way back towards the heart, every time it gets past the valve, it can kind of relax a little bit because it's not going to slide back down, right? So the valves are kind of a step ladder to make sure it keeps progressing back towards the heart and doesn't end up always down in your feet when you're standing up, right? There's other mechanisms that help with it as well, but those valves are only found in veins. It's nice that they both start with V. And those help to escort the blood back to the heart, okay? There's so much pressure in the arteries, they don't need valves. It's like, well, here comes some more. Get out of the way, get out of the way, get out of the way. Whereas in the veins, it's like anything we can do to help get it back to the heart. Okay? Okay. So this slide here just talks about the 3 layers, 2 of the 3 layers that I already mentioned. Okay? It just goes through a little bit of what the tissues are made out of. The inner one is the inner lining, so it's going to be epithelium. That's covering and lining. It's going to be simple squamous epithelium lining it. It's a nice slick surface so that the blood can shoot through, right? Tunica media is our smooth muscle, but it also can have elastic fibers, more so in our arteries versus our veins, right? And because it's muscle, it's going to control vasoconstriction and vasodilation. Vaso, remember, means vessel, not vein, but vessel. So arteries and veins, any blood vessel that has that smooth muscle layer will be able to vasoconstrict and make the diameter smaller, inner diameter, or dilate and make it bigger. And that's going to affect blood flow and blood pressure, right? So if you have a very, very small straw, the fluid is not going to be able to get through it very quickly. If you've ever tried to like drink a milkshake or something like that with a small diameter straw, you can't do it. Or it's really hard, right? Your eyes pop out of your head and stuff like that. So you have to have a bigger diameter in order to make things go through a little bit easier. So we can vasoconstrict and vasodilate to affect blood pressure and blood flow. Tunica externa is mainly connective tissue, collagen fibers out there, etc. That comes into play when we were talking about blood clotting, right? If there's a broken blood vessel, that intrinsic blood clotting that occurs starts out because those blood cells and those platelets get exposed to collagen fibers. They're seeing the outer layer of that broken blood vessel. But the collagen fibers help to give it a structure, right? They give it an outside layer to that. Okay. So we have now compared 2 out of our 3 main categories, right? So we talked about arteries and veins. I'm just going to bring you back 1 second to our capillaries. Look at the arteries and veins. We had 1, 2, 3 layers to the wall, right? 1, 2, 3 layers to the wall. How many layers do we have in a capillary? Basically 1. There is an endothelium, simple squamous endothelium on the inside like the others, and then it has like a basement membrane. That's it. There's no smooth muscle, there's no collagen fibers, etc. And the reason is that we said that these are exchange vessels. If we're going to have things going across the wall, we need a super thin wall. If we had all those layers, how are we going to get all these things to cross those layers? It would take forever, right? Diffusion wouldn't occur very easily. So we have a super thin wall in our capillaries, which facilitates the movement of these things into and out of the capillaries, the exchange. So we'll see that coming up as well. But super thin walls, not all 3 tunics in our capillaries. Okay, so now what we're going to do is we're going to branch out. That's kind of like a blood vessel joke. We're going to branch out from there, not just arteries and veins and capillaries. We're going to start looking at some of those other subcategories. So when these blood vessels branch and they split and they get smaller, they're going to change in their structure as well. So we're going to go from arteries to arterioles to capillaries to venules to veins. And we're also going to hit some of those other subcategories, elastic arteries, muscular arteries, things like that. Okay? And each time we get to one, we're going to say, okay, why do we have to have another name for this blood vessel? Why is that significant? There's got to be something different about it. We're going to call it an arteriole rather than an artery. Why? What's an elastic artery versus a muscular artery? Why? Why do we have to have those? So we're going to talk about the significance of them. So the first set of arteries that we have are called elastic arteries. also known as conducting arteries. These are the biggest ones that you're going to have, like the aorta, right? That's an elastic artery, as well as a brachiocephalic artery. What are our other two branches off the aorta? 1, 2, 3. Subclavian, common carotid. Left subclavian, left common carotid. Those ones are the closest to the heart. Those ones are going to have the most pressure exerted on them from the ventricles contracting. So they need to have a huge amount of elastin fibers throughout the wall. Okay? So it's going to be on the inside, in the middle, on the outside, lots of elastin and elastic fibers so that they can balloon out temporarily and then have this elastic recoil so they don't just pop. Okay? So elastic arteries can withstand all of those blood pressure fluctuations because they have a lot of that elastic fiber. They tend to have a large lumen so that we can let the blood go through them as much as we can. And because of all of that stretch that they have there, an elastic recoil, they're sometimes called pressure reservoirs. So what that means is a reservoir, if we talk about a reservoir you have around here, like Lake Pleasant or something like that, why is it called a reservoir?
Because we made it.
So we definitely need it. What do we need in that case?
Did we make Lake Pleasant? I thought we made all of the lakes in Arizona. I thought they were all man-made, or.
Yeah, right? So most of them are basically reservoirs. Like we have water flow coming through, and then we dam it off, and it backs up, and it pools there. So we have it on reserve because we need that water. So we don't want to let it go and have to track it down in southern, Mexico or something like that and say, hey, can we have some of that water back? So off of the Colorado, if we dam it in certain places, it pools. Now it's like, oh, okay, we can use some of that and deliver it to neighborhoods when we need it and things like that. So it's a reservoir that allows us to kind of keep it on reserve because we need it. Okay? So a pressure reservoir says, oh, we need that blood pressure because that's what's going to push the blood around this whole circuit. And what happens is when the ventricles are contracting, now is that systole or diastole? You got a 50-50 shot. Systole, right? Systolic pressure, systole is when the ventricles are contracting. Diastole is the relaxation period and filling period, okay? All right. So when the ventricles are contracting, there's this whole tidal wave of new 70 milliliters of blood that's shooting into the aorta. So there's a new tidal wave just whoosh down there. So that's a high pressure situation. The walls stretch out, okay? When the ventricles are relaxing and filling, how much blood is going into these arteries? Like nothing, right? Because we're jumping and we're filling, it's like we're refilling a water balloon. We're like, hold on, right? We're refilling the ventricles, so there's no new blood that's going out the aorta. So do they just like collapse and like dry up and stuff like that? What happens is that during that period when we're in ventricle systole, the arteries ballooned out like this. When the ventricles are going through their relaxation and filling, the arteries, especially these elastic arteries, are doing their snapback. So all the blood that ballooned out like here, all that new blood snaps back and shoots down the tunnel like that, shoots down the artery. So that helps to maintain the blood pressure due to that arterial recoil like that. So the elastic arteries serve as pressure reservoirs because they have an extra bit of blood that when they snap back, they can push down the circuit to keep that pressure up. Now, it's not as high as when the ventricles are contracting, right? It's like 120/70 or whatever, but it's enough to keep pushing the blood around the loop, even though the ventricles are like, hold on, I'll be with you in a second, I'm just refilling, right? They're going to handle it in that period of time. So that's why those are called pressure reservoirs, and that's why we need all of those elastic fibers throughout. Okay. So elastic arteries have a lot of elastic fibers in their walls. Muscular arteries are after that. So if we continue moving on, and today as we get into blood vessels, right, when we were doing the heart, we're like, oh, okay, I just see the major blood vessels. Like we said, aorta and brachiocephalic artery and stuff like that. But now, as we get into today, we're looking at other blood vessels, right? So we're like, okay, so subclavian artery, as it goes into the armpit region, it becomes what, morning lab? Axillary artery. When it gets down next to the humerus, it becomes the what? Brachial. And then it splits and becomes a radial and the ulnar. right? So it's going to continue to split and get smaller and smaller. That's when we get now to muscular arteries instead, okay? As we get to smaller branches after the elastic arteries, these ones have less of our elastic fibers now. We're a little bit downstream. I always think of it like a raging river. When it's first leaving, there's a lot of power to it. It's really loud. Sometimes there's turbulence, right? It makes it really loud, like those valves closing like we talked about. But as it gets downstream, and as there's friction as it's dragging in the stream bed, it gets slower and calmer, right? That's happening as we go out these arteries as well. So we got past the elastic arteries. Now we're getting down to the muscular arteries. They still have a thick tunica media, but less of the elastic fibers. And the thick tunica media gives us more smooth muscle so that we can do vasoconstriction. Because as we get down to these, we want to start routing this blood to different places where we need it. So that's the funny thing to think about in terms of blood pressure. Like when you are measuring it on your future patients, you're going to be checking it in one spot, right? You're checking it in one spot, usually on the brachial artery over here. But I guarantee you the blood pressure is not the same in all different areas of the body. You're just saying, oh, that's a snapshot of the blood pressure in the body itself. But it's going to be different in different places because, whether you're standing or sitting, whether you're, if this limb is active, if I'm doing activity with this limb and not with this limb, all of these muscles need a lot more blood flow, right? So we've got to dilate some blood vessels over there to get more blood flow over there compared to over here. So we get this constriction and dilation that routes blood to where it needs to go. So if we think about like the water systems, like the canal systems going around town, like in Gilbert and stuff like that, if you've ever gone along them, sometimes when you get to like these little farms, they'll actually have little valves where they can open it up and flood their field as necessary if they're growing something up, and then they can turn that valve and close it off if they don't need it. Same thing when we start to get out to these muscular arteries and stuff like that. The thick tunica media helps to constrict or dilate to route blood where it needs to go or not. It doesn't totally cut it off. We don't want that. But we can dilate it and let more blood flow or constrict it and restrict the blood flow and decrease the blood flow as necessary. Okay? So that's what our muscular or distributing arteries are doing as well, kind of when we get to our arterioles. So the difference between a muscular artery and an arterial is going to be kind of the size of it. So these are still going to be pretty big, like we said, like, radial artery and ulnar artery and stuff like that. Arterioles are much smaller, right? We're getting more and more and more branches. Think of a branching tree. As you get out towards the tip, you get more and more branches, but they get smaller as well, right? So like the trunk of the tree, like the pulmonary trunk, one. Then it splits into pulmonary arteries, like a tree branching that way. But then it branches and branches and branches, right? So the arterioles are going to be the smallest arteries that still have all three of our different layers, tunica intima and media and externa. But their major role, since they're the last arteries before the capillaries, is to protect those really delicate capillaries downstream. Because when we get to the capillaries downstream, how many different layers do a capillary wall have? One, right? Super thin so that we can exchange, super delicate though. So if we had that pulsing blood arriving at the capillaries, they would just basically explode. And sometimes they do. So we have to protect them at all costs. And so the arterioles control flow into the capillary beds and even though the blood vessel itself is much smaller than a muscular artery, arterioles have all three layers and they use that relatively thick tunica media to regulate the flow into the capillaries and make it nice and slow and easy as it arrives at the capillaries. So in fact, at the arterioles, this is where we stop having all of that pulsing. Previous to this, we see it extend out and then snap back, and extend out and snap back, like we do when we take a pulse, right? You take a pulse on an artery, you can actually feel that systolic and diastolic pressure causing it to change diameter. When we get down to an arteriole down at the bottom here, it's a small vessel. It has all three layers, but you can see that the middle layer, so here, right, they're showing us the relative amount of endothelium, how much elastic tissue they have, how thick the smooth muscle is, and how thick the outer fibrous layer is, okay? You can see, as we're looking at these, elastic arteries have a lot of elastic in their wall, by definition. Muscular arteries have less of that, and they have more muscle, right? And then when we get down to our arterioles, they're relatively small, right? We're on micrometers now instead of millimeters versus centimeters. But they still, the thickest layer is going to be that muscular layer. So their middle layer, which allows them to constrict and dilate, is still the thickest. And what it does is it stops all that pulsing nonsense. Because if we allowed that pulsing, higher pressure blood to arrive at the capillaries, it would just blow them out. So the arterioles are protecting the capillaries, taking away that pulsing sensation, and slowing down the blood flow and slowing down the blood pressure to allow it to get to the capillaries, right? When we have those little babbling brooks where it's going really, really slow and peaceful-like, that's how we want it in the capillaries, okay? So that's their main job. It's like big brother, big sister protecting the younger sibling, who's a little bit more delicate, right? Okay. So, elastic arteries, muscular arteries, arterioles. Now we're getting to capillaries. Questions yet? Yeah.
What's the precapillary sphincters? Are those the things that slow it down for the capillaries?
That's a good question as well. So we'll see some anatomy coming up. We know a bit of the anatomy of the capillaries already. Super thin wall, right? Epithelium on the inside, that's about it. We're going to find that there are these, what are called, by the name, precapillary sphincter. So the sphincter is going to be organized in a circle, kind of like when we think about like orbicularis oculi and orbicularis oris and stuff like that. When it constricts, right, it's going to then make it smaller on the inside. So there are bundles of smooth muscle that can, again, like those valves we were talking about, restrict the flow of blood into capillary beds or not, depending on the needs of that tissue at the time. So arterioles are definitely going to do that, but they're all about just kind of reducing overall pressure. The precapillary sphincters will very, very significantly restrict flow into a capillary bed or not. It doesn't have to go into all capillaries at all times. It depends on the needs of the tissue. So if it's really active, we want to flood that field. If it's not very active, we can kind of pass through it, but not go to all of the different capillary branches. So we'll see those coming up. Okay, so we talked about this already. Smallest and most numerous blood vessels. These are like the tips of the branches of the tree. And think about that too, right? So if you have a tree, trees and vascular plants, we're hearkening back to your general biology days, vascular, they have vessels in them, right? Vessels that are conducting things like fluids and nutrients, etc., around the body of the tree. When you go to those branches of the tree, when you reach the very tips, you get to the leaves, unless you're a cactus. We'll talk about that, I guess. But for a regular tree, you get to the leaves, and what happens to the leaves? The leaves are where exchange occurs, right? It's where you exchange oxygen for carbon dioxide and stuff like that. Even fluids leave the trees at that point, right? You get transpiration, you get the fluid that was there actually turns into a gas, and you get water vapor that goes into the atmosphere as part of the water cycle, right? The exchange occurs at the leaves. The exchange in the body occurs down here at these capillaries. So we're getting exchanges of all kinds of things. These are very, very numerous and very small, oftentimes only allowing one single red blood cell to pass through at a time. And they're allowing the exchange of respiratory gases, waste products, nutrients, ions, fluids, all the things that we said they're found in the blood that are important for distributing around. This is where we're exchanging. They're getting across the wall, okay? Now here's an interesting thing about it. We do have these things, the word is kind of interesting. That's a parasite. You're like, what? That's a bad thing, right? Have a parasite? So these sound the same, spelled different. Parasites are different because these capillaries are so very delicate. Parasites are the stem cells that go around the capillary wall, and they help to control what's called permeability, how much things are able to exchange, how leaky they may be at a certain time or not, right? So when we were talking about, we'll talk about it later, like inflammation and things like that, where things are allowed to leak into the tissues that may be infected or have damage. These will help to regulate how much exchange will occur at these capillaries. But the capillaries are basically everywhere. Almost every cell in the body has a capillary next to it. There are very few, there are some avascular tissues in the body. Epithelial tissues, avascular, right? Blood vessels go right up underneath it, don't go into it. There are certain types, right, if you talk about cartilages, stuff like that, avascular, they don't repair very well, things like that. But most cells in the body need a direct line of nutrients and exchange and things like that. So most cells in the body have capillaries going right next to them. Capillaries are so delicate and they're so numerous, you grow and reabsorb these. You form these and reform these all the time. All the time. Arteries and veins, really complicated. All the layers and stuff like that, you don't form those all the time. But capillaries, very delicate, you do. So if you are working out a lot or something like that all of a sudden, and those tissues have more demands, we're like, gosh, well, we got to get more blood there, you will grow more diverse capillary beds into that tissue to help route that blood there, etc. That even counts if, for instance, it's like wintertime and you've accumulated your layer of winter fat. You have added cells to your body, right? Cells require nutrients, oxygen, blah, blah, blah. If there are more cells, we got to have more capillaries to serve them. So in fact, when you gain fat, some little gains of fat, the cells, the adipocytes, the fat cells that you saw in 201, they just get bigger and smaller as they're storing triglycerides. But if you gain more, you have to make more cells. Like these ones are full, we got to make more. It's like when you get storage units in Arizona, right? They're all over the place. So like, I got more stuff, I got to get another one. So you make more cells, and in fact, you grow more blood vessels to supply those cells. So that also contributes to why sometimes it's hard to lose fat, because you made more cells. Now they're not really going away. You made more capillaries too. You made more tissue, and now you're trying to get rid of it. So it's tough. But capillaries, their main job is to route the stuff everywhere in the body as much as possible. Now, when we get to these exchange vessels, in fact, there are 3 subtypes of capillaries. So all of them do exchange. All of them have a thin wall. But there are gradations of that as well. So that we can, again, have things make it out or into the vessel easier or harder. I guess it's graded easy. It's all pretty easy, but there's easier still. So the top one that you see here are called continuous capillaries. So continuous capillaries are some of the most common. And these ones, again, we have our simple squamous epithelium forming the wall, and then a basement membrane. That's it. Okay? So we can find these like we're skeletal muscle, supplying the skeletal muscle cells, smooth muscle, in connective tissue, bones, narrow bones, stuff like that, brain and lungs. So those are our regular capillaries. The next ones are called fenestrated capillaries. So fenestrated capillaries, if we're comparing them, what's the difference? What do we see in that one? Holes. There are holes in the cells. So if you were doing an exchange in a continuous capillary, those things that are exchanged either have to go through those epithelial cells or they have to squeeze in between the cells. Well, in fenestrated capillaries, we actually have holes in the cells. Fenestrated means holy. So if you go to like French and stuff like that, the word for window is fenêtre. So a window, in fact, there's a word called defenestration. So if you were going along and you see somebody ahead of you and they flick a cigarette butt out of the window, horrible, really gets you irritated, but you're like, you're smoking it, why don't you just keep it in your car? Apparently you like the stink, you keep your own stink. Why are you throwing it out? Just saying. Anyway, throwing something out of a window is called defenestration because it's going out of a window. So it anchors you in the idea of these little holes are windows. With extra windows, it's even easier for things to cross in fenestrated capillaries. So we have holes in the epithelial cells, and it makes it easier for things to cross. So in places like the kidneys are constantly filtering and modifying the blood volume, the blood content, etc. So we want to make it easier for things to get across there. Small intestine. Small intestine is where we're absorbing nutrients that we've broken down in the digestive system. So we want to make it really easy to pull those nutrients into the body. What else do we have? Choroid plexus. What is that? You guys remember that? 201? So cerebrospinal fluid lining the ventricles, lateral ventricle, third ventricle, fourth ventricle, stuff like that. Choroid plexus is where we have these capillary beds where we filter out of the blood and we make cerebrospinal fluid that goes around the brain, around the spinal cord, etc. So we have to make that leaky as well. And then endocrine glands. Endocrine glands, what do endocrine glands release? Hormones, right? Hormones are bigger molecules. Bigger molecules, it's kind of hard to get them into the blood, so we want to have a leakier blood vessel like fenestrated ones instead. All right, finally, sinusoids. On the back of the heart, we saw a coronary sinus. Remember that? Right? So sinus and sinusoid is like bigger in terms of the capacity here. In this case, this looks like moths got to it or something, right? It looks chewed up. Not only the epithelial cells have holes, but the holes are bigger. And look, the basement membrane now is falling apart. So it is super easy to get across sinusoids because that allows a lot of exchange to occur. Exchange not only of molecules, but look at this. Liver, bone marrow, spleen. Why are those important in terms of blood? Right? When we talked about red blood cells, white blood cells, platelets, where they're born and where they go to die and stuff like that, those are the organs. So we're talking about whole blood cells having to go into and out of the bloodstream. You can't do that when we have a very solid wall. So we have big holes that allow these cells to be taken out of circulation, go into circulation, stuff like that. And then these are just a couple of endocrine glands, again, that are putting out tons of different hormones that make it easier to get in there. Okay? 3 different types of capillaries. So now we have 2 different types of arteries. We had arterioles. We have 3 different types of capillaries. We're up to 6 different blood vessels right now as different examples of things. Okay? Okay. So the capillary beds that we were talking about, they're, regardless of what type it is, they are all about exchange. They're all about flooding the field, like we said, if we're using that agricultural analog there, analogy. Right, we're going to flood the field, but we don't need to do that all the time. Just like the farmer doesn't need to flood the field all the time, only when things are actively growing. So in terms of regulating the flow into these capillary beds, here's our arterial. We said that's the last blood vessel before the capillary bed. And then here's our capillary bed. And then here's the venule where it's going to drain into, right? So if we have a very active tissue, it's going to need more fluid flow. Those cells are going to need to be served. And so we're going to need to flood that entire capillary bed. But if, let's say, it's a less active tissue, we don't have to be putting fluid through there all the time. We do, right, on a regular basis, but not all the time. So we have these precapillary sphincters, these bundles of smooth muscle that are surrounding some of these branches of the capillary bed. And so if we constrict those, then what happens is the blood still goes through the tissue, but it just kind of shunts through the middle and then goes into the venule, as opposed to distributing throughout this whole field. So in a tissue, we'll go back and forth. It's called vasomotion. It kind of allows it to flood, and then it kind of restricts it. And it allows it to flood, and it kind of restricts it. In a less active tissue, the amount of flooding is less frequent. In a really active tissue, we may leave it open the whole time those cells, like skeletal muscle cells, are really actively working. Okay? So this is also a way of controlling the flow distinctly into capillary beds. If we don't need to flood the field at the time, we can run it right through this central thoroughfare channel here. So the meta-arterial or the thoroughfare channel goes through the tissue, but it goes right through the center, if you will, and goes straight to the venule instead. Okay? So that's one of the ways that we can have that fluid flow controlled into these different tissues. Okay, so I'm just going to finish up. I see 10:43, 2 minutes. We have only 2 blood vessels left. We're working our way around. Arteries, arterioles, capillaries. Now it's venules and veins. Venules and veins are fairly easy. Venules are basically just small veins. They have the same 3 layers. They have thin walls. They have valves and stuff like that. But they are super thin in terms of their wall. So they have a very porous endothelium as well, like holes in it, so that some of our white blood cells can escape at the capillaries, but they can also escape into the tissues at the venules before we thicken up those walls and return back to the heart. So venules are just small, thin-walled veins. That's it. Okay? The veins themselves, when we compare it to the arteries, we said they have a thinner tunica media with less muscle. They don't have a lot of our elastic fibers in them, and they have these valves in them. You can see these are actual valves right there. And so that helps to escort the blood back to the heart. They serve as blood reservoirs. Because the pressure, the blood pressure and the blood flow is a little bit slower there, then at any given moment, if we're trying to see where's the blood in the body, a lot of it is sitting and moving slowly through the veins instead. So they're often called blood reservoirs. Now they do have smooth muscle, so if we constrict them, that also helps to squeeze it through, kind of like squeezing a toothpaste tube, right? If you squeeze the walls of a vein, it also helps to escort it back to the heart. But a lot of our blood ends up residing within our veins there. So they're called blood reservoirs instead, right? So you can see like 60% in our veins and venules, only about 15% in arteries and arterioles, and then various other places around the body. Okay? Okay. So I think this just kind of finishes up looking again at our capillaries, our thin-walled capillaries, our venules, and then our veins like that. So I think what we'll do at this point is stop right there, because we have now done our tour of our different blood vessels. We'll talk a little bit more about how the veins escort the blood back to the heart. But then we'll start, next time we'll go through a Q&A period at the beginning, and then we'll start looking at, again, blood pressure and blood flow, hemodynamics and stuff like that. Okay? All right.